this next section of notes is on mutations. A mutation is any change in the genetic code. And so a mutation could happen in either DNA or RNA. It could be a change in a base. It could be a change in a codon. It could be a larger change that affects an entire gene, or it could even be a change that affects an entire chromosome. So there are different kinds of mutations, and one is called a point mutation or a gene mutation. And this is a mutation of a base or codon of a gene. So of point or gene mutations, there are three main types. There are substitutions, and that's when we have one base that replaces or substitutes in for another. And since this is usually a change in one base, it could change one codon, which would mean that we could have one different amino acid in the protein sequence. Another type of point or gene mutation is an insertion, and this is when a base is added in somewhere in the sequence. And what this does is it pushes the rest of the bases over to make room for this one that's being inserted. And a third kind of point or gene mutation is a deletion. And a deletion is when a base is removed from the sequence. And when this happens, the rest of the bases will move closer together to fill in the gap of that missing base. So those are the three main types of gene or point mutations. There is a fourth one, and the fourth one is called a frame shift mutation. A frame shift mutation is a mutation that's caused by an insertion or a deletion. When we either insert an extra base or we take away a base, what that does is it shifts the codon reading frame. So how the codons are read becomes different because it alters those groups of three. And so what can happen is that an entire amino acid sequence could be disrupted from that point onward. So here's just showing the three main point mutations: insertion, deletion, and substitution. So here's an example showing what the correct mRNA should read, and then what the amino acids are based on that mRNA. It's then showing a substitution mutation. And you can see where the substitution occurred. It's in the third codon. A C was substituted instead of the G. What that did is it caused that one codon to code for a different amino acid. So it disrupted the amino acid pattern in that one location. Here's another example. Here we have an insertion which causes a frame shift mutation. So in the second codon, a C was inserted in. What that caused is all the other bases to be pushed downward. And so it completely alters the reading frame of what we should have. And so from the point of insertion onward, all the amino acids end up being different. Because an insertion and a deletion causes a frame shift and can alter so many amino acids, insertions and deletions, frame shifts, are usually more harmful than a substitution because we usually see more differences and more changes in the genetic code. An example of a point mutation that affects humans is sickle cell mutation. And you can watch this animation to get more information about sickle cell and the mutation that leads to it. And so then that leads us into a second kind of mutation, which are the chromosomal mutations. And these are large mutations that involve an entire chromosome. So these are usually more harmful than a point mutation because we're affecting many genes on a chromosome. And there are 4 different chromosomal mutations. So first we have a deletion, and in a deletion, a piece is broken off of a chromosome. If we're trying to make an analogy of a deletion mutation to a book, it would be like reading a book with pages missing. We might have page 1, page 2, and then all of a sudden page 3 is missing. And then it goes to page 4 and to page 5. Another kind of chromosomal mutation is a duplication. A duplication is when a piece is repeated on a chromosome. So we have the same information showing up twice. Oftentimes, this is from a deletion on one homologous chromosome that then gets added on to another. So if we're using our book analogy, this would be like having the same page repeated more than once. So it'd be like having page 1, page 2, page 3, and then page 3 again, page 4, page 5. An inversion is when we have a piece of a chromosome that is in reverse order. So in terms of our book analogy, it would be like having the pages mixed up and in the wrong order. So we could have page 1, and then page 5, page 4, page 3, page 2, and then page 6. So that span from pages 2 through 5 ended up being in reverse order. And then finally, the 4th kind of chromosomal mutation is a translocation. And this is when a piece is added from a completely different chromosome. And so this is oftentimes from a deletion on a different chromosome. So in terms of our book analogy, this would be like reading a Harry Potter book, and then in the middle of the book, all of a sudden, it switches to Twilight, a completely different book within the book you were already reading. And so these book analogies give you an idea of how these mutations would affect an organism, where the information may be missing, or it may be there twice, or it may be in the wrong order, or it might not be where it's supposed to be. And that can cause lots of problems for the organism. Now, there are different causes to mutations. And one cause would be an external factor that causes a change in the DNA. When that happens, that external factor is called a mutagen. That's usually some environmental influence that permanently changes the genetic material. And here's some examples. Radiation, like ultraviolet radiation from the sun, X-rays, different chemicals, even different infectious agents like viruses and bacteria, all have the potential to mutate an organism's DNA. Other causes would be internal factors, and these would be random errors that occur in replication or protein synthesis. Now, these internal factors, it makes a difference where the mutation occurs. If it occurs in a gamete, that mutation can be passed on to the offspring. If the mutation occurs in a somatic cell, that will affect the individual, but it won't be passed on to offspring. And finally, the outcomes that mutations can cause. It is possible for mutations to be neutral or silent, which means there's a change in the DNA or the RNA, or there's some change in the genetic material, but it causes no effect on the organism. It doesn't change the amino acids. And this animation shows that we could have CUU, which means leucine, and even if it mutates to CUC, that's still going to give us leucine. So we would have a mutation, a change in the genetic code, but it's not going to cause any effect on the organism. We're still going to create the same protein. Outcomes could be harmful, And a harmful mutation would disrupt the protein and normal biological activities of an organism. And sometimes mutations can be helpful. A helpful mutation gives that species genetic variability and it offers individuals some sort of a survival advantage.